Okay, welcome everyone to this lecture on the use of organoids in the analysis of neural stem cells. So I'm sure everyone here will agree that the human brain is the most fascinating organ that nature has generated. It's also the most complex organ, and unfortunately, this complexity comes at a price. Our brain is the home of some of the most devastating human disorders like Parkinson's, dementia, schizophrenia, depression, epilepsy. And it's thought that about 25% of the entire population at some point during their life suffers from a brain disease. And while this is a huge medical problem, the treatments that we have available are not adequate for this desperate situation. And there's many reasons for this, but one of them is that research on the development of disease treatments has mostly been done by in animal experiments. And while animal experiments are necessary and they've taught us a lot about how our brain develops, they have certain limitations. And one of them is shown here. So this is a patient who suffers from severe microcephaly. The patient carries a mutation in a gene that is called NDE1. NDE1 is a microtubule binding protein that is conserved all the way from yeast to humans. But when you make the corresponding mutation in a mouse, not very much will happen. And there's many examples like this. Very often, mutations that cause devastating brain disorders in humans have only very mild consequences in rodents. And there's a number of reasons for this, but the key reason is that our brain is not just a blown up mouse brain. And I would like to remind everyone that during brain development, the number of neurons that constitute an entire mouse brain are generated in a human fetus every 4 hours for 9 months. But in addition, there are processes that are happening during human brain development that you do not find in rodents. And some of them will be shown on the next couple of slides. So the neurogenesis in our brain starts from neural progenitor cells that are also called radial glia cells. And initially during brain development, these cells divide symmetrically. So one cell generates 2, 4, 8, 16, 32, and so on. And this leads to an exponential expansion of the progenitor pool. And at some point, these progenitors switch to an asymmetric division mode, where one progenitor generates one progenitor and one cell that migrates out and becomes a neuron. Or this other cell can become a transit amplifying cell, a so-called intermediate progenitor, which will then generate 2 neurons that migrate out and wire up. Now, this initial phase of symmetric division lasts much longer in humans. This is a rat brain and a human brain at a comparable stage of development, blown up to a similar size. And while the more posterior parts are very similar, you can see that the cortex is vastly expanded in humans. And this is because many more of these symmetric divisions have taken place. In addition, there are progenitors that exist in primates and humans, but do not exist in rodents. So in the mouse, per progenitor division, the progenitor cells generate one transit amplifying cell, which divides once more to generate 2 neurons. In humans, however, they can generate so-called outer radial glia cells, which continue to divide asymmetrically, generating hundreds of intermediate progenitors and hundreds of neurons. And these outer radial glia cells form an additional layer that is characteristic to the human and to the rodent brain, and is not found in, to the human and to the primate brain, and is not found in rodents. So this is good news because it tells us why our brain is so much more complex than that of a mouse. But it's also bad news because it means there are certain processes that we cannot study in rodents. But fortunately, there is hope. We're living at a time where there's a complete revolution in the way we do biomedical research. And that's because 4 technologies have recently been developed. The first one is human genome sequencing, We now have thousands, soon millions of complete human genomes. Reprogramming is the second one. Thanks to the methodology developed by Shinya Yamanaka, we can generate pluripotent stem cells from each and every patient. The third one is we can then use these pluripotent stem cells and generate all kinds of three-dimensional cell cultures, so-called organoids, that allow us to analyze development and disease in vitro. And the fourth one is we can then manipulate the genome and write in the genome, like in a typewriter, using CRISPR-Cas technology, and study the consequences of this in vitro. And today, I would like to focus on one of these. I would like to focus on organoids. And before I do that, I want to give you a little bit of a historical background. Now, one of the pioneers of organoid biology is Johannes Holtfretter, and he was the first one to show that when you dissociate cells, in his case, he used amphibians, they will find each other, and the right cell types will will form clusters together. And in a most courageous experiment, what he did was he isolated ectoderm, endoderm, and also connective tissue cells and put them together. And then the cells sort out. So the ectoderm bulges out and the connective tissue cells are in between and form a three-dimensional cell culture that has a certain structure. And if you want, this is the first organoid that has been generated. Now, in the meantime, organoids have really gained a lot of attention. And the idea is basically that we can develop iPS cells from individual patients, which will then be allowed to differentiate. They will sort out. They will form a specific three-dimensional architecture and then build an organoid that one can then use to model disease, test drugs, and also eventually maybe to form organ replacements. And so on the next couple of slides, I would like to tell you something about what we know about organoids, how they are generated in general, what their historical roots are, and how we can generate organoids for particular areas of the human brain. But at the beginning of this, I would like to tell you what is the definition of an organoid. This definition, there are many definitions in the literature. This comes from the father of organoid biology, Hans Clevers, who wrote a review in 2016, where he defined an organoid as a three-dimensional structure that is grown from stem cells, that consists of organ-specific cell types, which self-organize through cell sorting and spatially restricted lineage commitment. And in addition, other people have also included that the organoid has to be able to fulfill at least a few of the functions of the specific organ. So number one, derived from stem cells. Number two, has the organ-specific cell types. Number three, they arrange in the correct way. And number four, the organoid performs at least some of the functions. Now, in the case of the brain organoids, organoid biology actually has a long tradition and is firmly rooted in a lot of two-dimensional cell culture that has been used to model brain development. Now, I'm sure many of you, all of you will know that our brain is developed from initially the inner cell mass, which then during gastrulation sets up the neural fold, which invaginates to form the neural tube. And on the anterior most part, there are vesicles that are forming, that are bulging out, and that form the different areas of the human brain. Now, the specification of the nervous system, what we know about that goes back to the classical experiments that were done by Hans Spemann and Hilde Mangold, who have identified an organizer that induces the formation of the nervous system. And they have done this again in frogs. And what this organizer, which is called the Scb&R organizer, what it does is it contains inhibitory activity. There are two kinds of signaling pathways. First, BMP signaling, and the second one, TGF-beta signaling, that are inhibiting the formation of neurons and the nervous system. So another way to look at this is neurons are the default pathway. If you give cells no information and you let them differentiate, they will turn into neurons. But endogenously, in most areas, BMP and TGF-beta signaling are active, and they prevent this and direct the ectoderm cells to the epidermis instead. And so what we learn from this is we need to inhibit BMP signaling and TGF-beta signaling, and this is done by adding noggin, a certain chemical, and an ALK inhibitor, SB31542, to the culture. And because both of these inhibit a signaling pathway that uses SMAD signaling, this is called the dual SMAD inhibition. And dual SMAD inhibition has been a classical way of deriving neural cell culture. And in 2 dimensions, the neural progenitors already form these rosette-like structures that can be plated. There have also been some early attempts to generate neural aggregates, so-called neurospheres, which also can be derived from ES cells. And the cells already arrange themselves in 2D. But the key for generating 3-dimensional tissue goes back to a Japanese scientist who is called Yoshiki Sasai, and unfortunately is no longer with us. And together with his postdoc, Mototsugu Eiraku, he developed a method that is called the SFEBQ method. Basically, the idea is that one takes embryonic stem cells, human embryonic stem cells, and places them in a 96-well plate, let them sink down, and they will slowly aggregate and form balls of tissue. Here is a movie of how this process looks like. So these are the ES cells at the bottom of the well, and they form these balls of tissue. And these balls will become 3-dimensional, and this is the start of the formation of a brain organoid. So Yoshiki Sasai became quite famous in generating organoids for the retina, but also for the brain, initially in mouse, but then in one experiment also in humans. But then it was work from Madeleine Lancaster in my lab that actually showed that we can use organoids to model human disease. And the method that she developed is shown in this movie. So we start with colonies of embryonic stem cells or iPS cells, which we then dissociate by limited protease digestion. Then we take between 5,000 and 8,000 of those cells, put them into a 96-well plate. This is the SFEBQ method. They will go down, they will coagulate, form what's called an embryoid body. We then change the media to push them along neuroectoderm fate. And then we place these balls of neuroectoderm into a substance that is called matrigel. Matrigel is a collagenous substance that mimics extracellular matrix, and the organoids grow inside the matrigel on an orbital shaker. And what will happen is that inside the organoid, the cells arrange themselves in a way that is very similar to a human cortex. They form a cortical plate where they divide symmetrically, then asymmetrically, the neurons will migrate out. They form an additional layer, which is called the cortical plate. And some of the first organoids that we have generated look like this, where here on the most anterior part, you find a ventricular zone. Here is the lateral ventricle. In red are the progenitor cells. In green are the differentiating neurons. And the cytoarchitecture here is not too dissimilar from that of a fetal human brain. Down here is the choroid plexus. This is the area that generates cerebrospinal fluid. And here are other brain areas. This is again a schematic of how we recapitulate human brain development using this methodology. So induced pluripotent stem cells are first used, then a few thousand of those are placed into a 96 well plate. They form an embryoid body, which is then subjected to neural induction so that you form these balls of neuroectoderm. Then they're placed into matrigel, and the organoids form these buds. Each of these buds is a little small human cortex. And if you then let them grow inside the matrigel, they generate these so-called cerebral organoids, which we can then grow for a very, very long times. I'll show you later experiments where we've grown them for over 1 year. And we can also show that even human developmental processes that occur at that time can be recapitulated. So this is how organoids really look like. Here they're actually in a spinning bioreactor, but you can also culture them in a 6-centimeter dish. They're these intransparent balls of tissue. This is an example of an organoid that actually grows a human eye. It's a very rare event, but it does occur every now and then with particular cell lines. And when you make sections through these organoids, they, in this early stage, they really look very much like a real human fetus. But what is really remarkable about the organoids is that they generate neurons, and these neurons then form functional neuronal networks. So on the movie that I will show you on the next slide, we have made an organoid transparent, and we have labeled only 1% of the cells fluorescently, so you can appreciate the neural networks that they form. So in the movie that I will show you, the progenitor cells are red and the neurons are white. And here you see the progenitor cell layer, and now you see the neurons, and you can see they form these processes, and they form very, very complex neural networks. But what is most important is that we can actually use the organoids to model neural developmental disorders. So the first disorder that was modeled in an organoid, in a brain organoid, is called microcephaly. So we teamed up with Andrew Jackson, a pediatrician neurologist in Edinburgh, who provided us with fibroblasts from a microcephaly patient. We reprogrammed those and then grew organoids. As I told you before, patients who suffer from microcephaly have a severely reduced brain volume, and we can recapitulate this phenotype in the organoid. The patient-derived organoid is much smaller than the healthy organoid. And now what we can do is we can ask, why is this brain so much smaller? First thing we did was we sequenced the genome, and we found that the patient carries a mutation in a gene that is called CDK5-RAP2. To find out whether this mutation is responsible for the phenotype, we repaired the mutation using CRISPR-Cas technology. And what you can see is this is the healthy organoid, this is the patient organoid, and this is an organoid in which we have repaired the mutation. And what you can see is that the size of the organ is almost completely recovered. And through a whole lot of experiments that I don't have the time to show you, we could show that CDK5-RAP2 is actually a centrosomal protein, and it is required for the correct orientation of the mitotic spindle and the correct orientation of the cleavage plane. And what happens in those patients is that the progenitor cells cannot maintain the correct orientation of cell division. And for that reason, they start dividing asymmetrically far too early, and they begin to generate neurons far too early, and they do not expand in the same way. So this process, this extended symmetric division that I have described to you before, is absolutely essential for the human brain to reach its size, and we could gain some insight into where the disease comes from. Now, unfortunately, microcephaly is a severe disorder, and once the brain has reached its size, it cannot be rescued. So this was an interesting proof of concept experiment, but it doesn't really hold any potential for therapy. But organoids entered became really famous when they were used medically for the first time. And this was in 2015, when the last time the World Health Organization called for a global state of emergency, the last time before the SARS-CoV-2 virus. And this is because at that time, the Summer Olympics were planned in Brazil. And in Brazil, a certain virus that is called the Zika virus was identified and spread enormously. This virus comes originally from Africa, and then through various African countries, and then Indonesia and French Polynesia, it started to reach Brazil, and it spread throughout the country and also into Mexico and the southern part of the United States. Now, the Zika virus per se is not very dangerous. It causes a very light flu, but it was observed that at the time of this epidemics, there was an enormous increase in the frequency of microcephaly. And many women who were infected with the Zika virus during pregnancy actually gave rise to children who suffered from severe microcephaly. And so Guo Liming and Hong Jung Song teamed up and used our organoid methodology to model Zika virus infection. And this is a normal organoid, and this is an organoid that's infected with the Zika virus. And what you can see is that it is much smaller. And through a whole lot of experiments that I don't have the time to show you, it was demonstrated that the Zika virus specifically infects only progenitor cells when they are dividing. And this is also why fetal brains are very susceptible, whereas adult brains are not, because in adult brains, there's only neurons and the virus cannot infect neurons. But in the fetal brain, when the progenitor cells divide, the virus is inherited by the neurons, and it will then kill them over time. And the massive cell death that is happening in those neurons is responsible for the phenotype. My own lab also then started to work with the Zika virus, and we showed that we can use organoids to distinguish different modes of viruses that are causing microcephaly. We compared the Zika virus with herpes simplex virus and also cytomegalovirus. And for those of you who are interested, you can read this up in this cell stem cell paper. Now, the original organoids that were generated were modeling the human forebrain, but since then, a whole lot of different protocols have been developed to generate essentially any part of the human brain. Now, I don't want to go into the details of this. Here is a review that summarizes the various different organoid methodologies. Basically, in all of these protocols, what is done is that the endogenous patterning is modeled. And there's a few patterning pathways that are important. For example, sonic hedgehog is inducing ventral freight throughout the brain development. The wind signaling is inducing caudal patterning. And varicaudal, like spinal cord fate, is induced by adding retinoic acid. And there's another morphogen called FGF8, which can induce the patterning along the midbrain, hindbrain boundary, but it also is used to model very, very anterior development. So I'm just telling you this to illustrate the basic principle of this. In organoid protocols, the endogenous signaling pathways are recapitulated, and this is the basic principle that's underlying all these different cocktails of growth vectors that are used to generate different organoid types. So we can model basically any part of the human brain, but there's even more complex technologies. So the cerebral organoids that I showed you before model the development of many different parts of the brain because they are using minimal guidance and minimal influence through growth factors. But with patterning factors, you can generate, for example, pure dorsal organoids or pure ventral organoids. What you then can do is you can fuse these organoids together, and the interactions that are happening between different brain parts will also be recapitulated. The most modern way of making organoids is axially patterned organoids, where a morphogen is provided in a graded manner so that the polarity of the brain is recapitulated. And then there are other more modern technologies where organoids are co-cultured with other cell types, for example, microglia or endothelia, to model vascularization or the development of an immune system. So I just show you the different principles of these various methodologies, but now I would like to give you a few examples for how these types of technologies can be successfully used. I would like to start by showing you dorsal-ventral fusion organoids. Why is this important? This is important because the neurons in our cortex come in two classes. There is excitatory neurons, and these are the ones that develop along the lines that I have shown you before, but there's a massive number of inhibitory interneurons. These come from the ventral, from the subpallial parts of the developing brain, and then will migrate tangentially to the dorsal part where they integrate into the circuits. And it's this interplay between excitatory and inhibitory neurogenesis that is absolutely crucial for the development of the cortex. And so the way we have recapitulated this, and it's also the lab of Sergio Pasca that has developed this method at the same time, is we generate a dorsal organoid that is generated by using the hedgehog inhibitor cyclopamine A, or a ventral organoid that's generated by using a Wnt inhibitor, IWP2, and an agonist for the sonic hedgehog pathway. And so pure dorsal, pure ventral are put together into one droplet of matrigel, and over time, they completely fuse together. And what you can see here in this fusion, where we have labeled interneurons only on the ventral part, but not on the dorsal part, is that these interneurons will then migrate into the dorsal part. And so this is a very beautiful model system for modeling interneuron migration. And I would like to just show you this movie of a migrating human interneuron, because I believe this is one of the most beautiful cell biological processes that are actually happening during brain development. So these interneurons are sending out these long processes which try to explore their environment and then make decisions as to where to go. And then the cell body, which is actually here, is then dragging along into that direction. So assembloids can be used, so these fusion organoids are now called assembloids, and these assembloids can be used to model neural migration or the outgrowth of axons in a certain direction. But organoids can also be used to recapitulate human-specific disease mechanisms. And this is work that we have done about 2 years ago, where we modeled a disease that's called tuberous sclerosis. Tuberous sclerosis is a disease that arises from mutations in TSC2, which is an inhibitor of mTOR signaling, and an increase in mTOR signaling then causes the formation of benign tumors, and it also causes the formation of these so-called tubers that one can see in the MRI, and that consist of enlarged giant neurons. We made an organoid model for TSC, and depending on the way we culture the organoids, they either form these new tumors on the surface of the organoid, or they form these giant neurons inside. So we can completely recapitulate the disease phenotype. And we could then use this to show that TSC is generated from heterozygous cells in humans, whereas in mice, you need to make the mutation homozygous. And we use single-cell transcriptomics to see whether there are any cell types that are only developing in these mutant organoids, here in this UMAP plot, the healthy cells are shown in orange and the mutant cells are shown in blue. And you can see there are certain interneurons that only occur in the TSC mutant organoids. And there's also certain type of progenitor cells. And by carefully comparing the markers that are expressed in those various cells, we could show that these progenitor cells come from a region that is called the caudal ganglionic eminence, and they divide very late in gestation. And so we call them caudal late interneuron progenitors, or clip cells. And through a whole series of experiments, we could demonstrate that what happens in TSC patients is there are tumors that are developing from the overproliferating clip cells, which overproliferate because they have more mTOR signaling. And then they generate interneurons, which then migrate into the cortex, and these generate the tubers. In mice, the clip cells do not exist. And that's why making the same mutation does not cause the same disease phenotype in mice. And so we're very excited about this for two reasons. First of all, because we've generated a disease model that can now be used to test certain compounds for their effectiveness, but also because we have found a new cell type that is very important for human brain development, but doesn't exist in rodents. And so we wanted to know what's the endogenous function of this cell type. And for that, I just need to give you a little bit of background. Work from the lab of Arturo Alvarez Buella has shown that in the human brain, there are certain cells that after birth, there's a massive migration of interneurons into the cortex. So I showed you before that interneuron migration is important for brain development. But in mice, it is finished at some point during brain development, and interneurons are only migrating into the olfactory bulb, but not in the cortex. But in humans, there's a massive cortical interneuron migration. And Saku Wong in my lab was able to recapitulate this process by growing organoids for 400 days. And she can even make live imaging of this very interesting process. I've shown you before how embryonic interneurons are migrating, and you will probably see that these interneurons migrate in a very, very different way. There's clusters of these interneurons that glue together and migrate into the cortex. And so we're very excited that organoids can actually be used to model processes that are actually happening in the human brain after birth. And these cells that are migrating express certain markers of the clip cells, raising the possibility that the clip cells or a subpopulation of those clip cells may actually be the ones that are responsible for this postnatal process. And of course, if you put all of this together, what you could say is that the human brain, in order to maintain plasticity of the cortex until after birth, it needed to prolong interneuron migration, but this makes us more vulnerable to certain neurodevelopmental disorders. So basically, the price we pay for the complexity of our brain is that we are more vulnerable. But actually, what our brain does is it fires action potential. It has neural activity. And what I wanted to show you on the next couple of slides is that we can also use organoids to study neural network activity. So this is a movie of an organoid expressing GCAM, a calcium indicator. So basically, whenever the cells flash, they are active. And what you can see is that the entire network shows these bursts of activity where the whole network is firing like now. We can also do electrophysiology in the organoids, and then we can again see these bursts of activity. And the good thing about electrophysiology is that you have a much, much better temporal resolution. And what you can see is that what looks like a uniform burst of activity in electrophysiology actually looks like a very complex pattern of activity that looks very different in the beginning than in the end. And so we wanted to ask, can we use this to model the neural network activity defects that accompany tuberous sclerosis? And for this, we teamed up with a group of clinicians in Utrecht who work on tuberous sclerosis. They perform surgery on patients, and during the surgery, they place electrodes directly onto the brain of the patients. This methodology is called ECOG, and it leads to recordings like this. And what you can do with the neural network activity that you measure is you put a certain bandpass filter over it. So you measure activity only, in this case, between 80 and 250 hertz. And then you really see the difference. This is a healthy electrode. So, for example, one that was very far away from the epileptogenic core, and it records this very small activity, but a long time period. Electrodes that are close to the epileptogenic core record a much higher amplitude activity, but the bursts are much shorter, that you can see. And so the surgeons have defined certain patterns that they use to define pathological activity versus healthy activity. And based on this, they make the decision on what part of the brain to cut out, and also whether their surgery was successful. So we did the same thing in organoids, and this is really a very small snapshot onto a huge number of experiments where we've compared a TSC patient organoid with an isogenic repair. And again, you can see that the oscillations have a much higher amplitude and they're much shorter. And so we can take all these quantitative indications in vivo, the duration is shorter, the amplitude is higher, the frequency higher, power higher, entropy higher, and there's a certain coupling between these high frequency oscillations and low frequency oscillations. And each and every one of these features can be recapitulated in an organoid. So I'm showing you this because this describes really the limit of where organoids stand at the moment. We're beginning to model neural disorders, not only on their cell fate level, but actually on the circuit level. We're beginning to understand the neural network activity changes that occur in organoids in pathological situations. And of course, this is a very, very interesting inroad into pharmaceutical development, because these are the things that you would like to change with a successful drug that is anti-epileptogenic. So the last thing I wanted to show you is that we can use organoids to build complete human brain circuits. And the circuit that we decided to recapitulate is the so-called dopaminergic reward system. So dopaminergic reward system is the circuit that fires when you have done something that makes you proud, something that you wanted, something that generates positive emotions. And unfortunately, one can also artificially stimulate that circuit. Cocaine is very good at this, and therefore it's also a major site of addiction, but it's also the site of habitual addiction. For example, gambling or these things act on that circuit. And unfortunately, it's also the site of a major neurodegenerative disorder. In Parkinson's disease, the dopaminergic neurons are degenerating, and this explains the symptoms. And for this reason, the dopaminergic circuit is one of the most deeply analyzed brain circuits for pharmacological intervention. And we wanted to recapitulate this circuit. It contains 3 parts. It starts here in the so-called ventral midbrain, where there are dopaminergic neurons that are created there. They project on one hand into the striatum, a deep part of the human brain, or they project into the cortex. And so we wanted to build these 3 parts and then put them together in a way so that they function as a circuit. This is the work of Daniel Reumann. And for those of you who are interested, you can read it up in this publication. So Daniel generated first ventral midbrain neurons. This marker expression shows you that we have generated the right cell type. He then generated striatum, and here the so-called medium spiny neurons are characteristic for the striatum. And of course, he used the cortical organoids that I've already described to you before. And in order to fuse them together, we 3D printed a mold that we could use to generate this PDMS form, where you can embed 3 organoids and they will touch each other, and ultimately they will grow together. And in this experiment, we've put GFP only into the midbrain part. And if you then focus here onto the striatum, you see that there are major axon bundles going through there. And if you focus on the cortex, you see there's also many axons that go all the way into the cortex. Now, I don't want to bore you with the details of this. We can also show that there's electrical activity going from the midbrain into the cortex. You can stimulate the neurons in the midbrain and record activity in the cortex. But I wanted to show you a movie of how these neurons actually develop. These are the axons that are coming from the midbrain into the cortex, and you can see how they're actually wiring up in the right way. But what we can also do is we can implant neurons into these organoids. So here we've implanted neurons that were labeled with GFP into the organoids, and you can see that they will find their way. And we can do 2 kinds of things with it. First of all, we can model cocaine addiction. You can treat these organoids with cocaine, and then the neural network activity is characteristically changing. But then you can withdraw the cocaine, and you can see that certain aspects, not all aspects, but some aspects of the changes are maintained. And that shows you that there are certain irreversible activities that cocaine is inducing. The second application is this can be used for testing cell therapy for Parkinson's disease. So you probably know that Parkinson's disease arises from a degeneration of the dopaminergic neurons, and there are currently 3 places where clinical trials are underway to generate these dopaminergic neurons in vitro and inject them directly into the brain of patients to see whether one can cure Parkinson's disease with that. And there's currently no way to test these the success of this, but we have obtained some of these neurons. We can inject them into the organoid, and we can now use this to test whether the individual batches of neurons are actually successful and how many neurons you actually need in order to repair a defect. So I've shown you that organoids can be used to generate assembloids, to look at neural migration, to model neural network activity, but also to recapitulate entire neuronal circuits. The final thing that I wanted to show you is that we can also, so organoids in vitro reach a certain degree of maturity, but they have no blood vessels and they have no immune system. In order to overcome this, the group of Rusty Gage and later also the group of Sergio Pasca has developed methodology to grow organoids in vitro and then implant them into the brain of rodent where they reach full maturity. And recent really exciting work from Sergio Pasca's lab has shown that actually if you stimulate the human neurons in these rodents, you can change the behavior of the mice. So by implanting human organoids into rodent brains, they will find their place and they will fully integrate, they will be vascularized, and they will participate in the neural network activity in the animal. So with this, I would like to close, but I would not want to stop without giving you a little bit of a perspective, because one of the challenges in the organoid field is that, for my taste, there's a few people out there who make promises with organoids that raise fear in the population, in the general public, and in my view are totally exaggerated. So I just wanted to say that organoids at the moment can model human-specific gene functions in brain development. We can also use them to model human-specific lineages and human-specific cell types. And we can model early developmental diseases that have strong phenotypes. What organoids will be able to do in the future is to model common neurodevelopmental disorders like autism or even schizophrenia, that they can model neural network activity changes and changes in the architecture of the neural networks that underlie the disease. But what organoids will never be able to do is to model higher order brain functions, to model things like behavior or consciousness, to model interhuman cognitive abilities. I believe that organoids from any healthy patient will look more or less the same, and differences in intelligence or personality will never be able to be recapitulated in an organoid. And then there's an entire field out there that is called biocomputing, where people use organoids and the neurons derived from there and put them together in a way so that it seems like they perform mathematical operations. Some of you may have seen this work where organoids start to play Pong. If you really go into the details of this work, it is correct and it's done quite well, but the way the organoids are seeded and the way the electrons are wired up, it is absolutely clear that this system, based on very, very simple neural feedback, will perform a task that then looks to us like the organoid has learned to play a computer game. But there is no there's no underlying specific task that one could not have predicted before. So with this, I hope I've been able to give you a little bit of an overview over the organoid field. Of course, I could only scratch the surface, and that's why I made this slide where there's a whole collection of recent reviews that those of you who are very interested in organoids and in their biology, that those of you will be able to analyze further. And with this, I thank you for listening to me, and I'll be happy to, if there are any questions, you can always send them either to Emanuela or to me. I'd be happy to engage in any conversation.